Is this already working? All right. Thank you. Sorry for the few minutes of delay. Some of us, I think, were teaching, including myself, until now, so we couldn't arrive any earlier. Thanks, Gisela, for being here today. Gisela Detrell is going to be talking about life on Mars or surviving on Mars. I think it's a quite appealing topic. Gisela does research, or the focus of her research is on life support systems and on human exploration and human habitability on Mars. So I think she's going to be talking about it for a while. She did aerospace engineering in Polytechnic University of Catalonia. Then she did a PhD between UPC and Stuttgart University, where she continued for a postdoc. And recently, well, a couple of years ago, she became associate professor at TU München, where she is leading now a group on these topics. So thanks very much again, Gisela. Floor is yours.
Thank you. Thank you for the invitation. I'm really happy to be here today. Coming from Barcelona, I've been many years now in Germany, but it's always nice to come back to Spain and get to explain what we're doing now from Munich on. So yeah, today's topic is surviving on Mars. So the first thing we'll do is check the conditions on Mars. And I'm try to be a positive person always, so I try to think what's the good thing we have on Mars. So we start with a couple of positive things. The day is longer. I'm quite sure we notice all. We need a couple of more minutes a day for sure. The year is double as long, so we are double as young on Mars. And gravity is 1/3. We can jump 3 times higher. So imagine a basketball tournament on Mars. Now, that's more the funny side of it. The reality is a little harsher than that. Temperatures on Mars are not made for us. There's very nice temperatures up to 20 degrees, which feels very comfortable, but it goes down to minus 100 several degrees, so absolutely too cold for us. We also have radiation here on Earth, as you probably all know, coming from the background you have. We are quite well protected with the magnetosphere from the space radiation, but on Mars we'll not have this magnetosphere, and radiation will be for us humans deadly. And same with the Mars atmosphere. It consists mostly of carbon dioxide, and we need oxygen to breathe as humans. So as you can see, again, not compatible. So Mars is definitely not made for us. It's not a place to be. With that information, who would like to come to Mars? No? Yeah, it is a crazy idea. But quite honestly, who would have thought 200 years ago that we humans can fly? And I just took a plane yesterday to come here to give you a talk without thinking much about it. In a similar way, 100 years ago, no one would have thought that we can live in space. And we have been there now continuously for more than 25 years. I don't know how old you are, guys, but it's likely, my students in my lectures, as long as they have lived, there have been humans not living on Earth up there. So it's quite an achievement. So why should it not be possible one day to also imagine that we could live on Mars, as hard as it is? So if the city on Mars would look like this, who would like to come? But it's a couple of people. Okay. So that was a kind of side project I had with several colleagues from several universities, worldwide, mostly Europeans. And we took a challenge. We met several times in informal environments and say, hey, we should work together at some point. It is a group of engineers, architects, scientists from different disciplines. And we all had the goal of, or the wish to design habitats outside of this Earth. Might be Mars, could be the Moon. And at some point we saw a competition from the Mars Society, which goal was to design a city on Mars for 1 million people. And now I'm still asking myself today, why would we send 1 million people to Mars to start with? But okay, that was a goal, that was defined. That was not for us to judge, but it was for us to think, how would a city look like? And I don't believe this is something we will see anytime soon. I'll definitely not see that in my lifespan. But for us, it was an exercise to see what are the challenges we would face one day if we would be able to do that. If we would reach a point where we've already been to Mars, we already had a couple of people there, a scientific base, what does it mean to go from this scientific base to a big city? A city, it's not something usually an aerospace engineer designs, so we have to think about the mobility between the different parts of the city. We have to think about how many hospitals do we need, which services does the city need. So it's a different way of thinking. And that helps us, such an exercise, to think which technologies, also from the space side, we need to develop to make that one day possible. So it's more a thinking exercise and evaluate which research areas do we want to start or focus on more today. So one day a city like this can be possible. And at the end, it's all about living in a harsh environment, as we have seen. Mars not made for us. But we are also living in harsh environments here on Earth. For many, many years, we have had now scientific bases on the Antarctic. That's the Neumayer station, the German station. We're also able to live in desert areas where water might be a very scarce resource and temperatures also vary, not as much as on Mars, but we also go from very high temperatures to very low. We're also able to live underwater with a submarine, which is pretty much like a space station underwater. And we're even able to go underwater on our own. That could be an astronaut on an EVA. We can do the same underwater for at least an hour, depending on the gas consumption and the number of bottles we take with us. But we're able to live on harsh environments already. Now, what do we need to survive? Because that's the point. It's not about making it a nice city or a comfortable environment, but the main point of today's talk is about surviving. So for surviving, you all know what you need to survive. We all take oxygen, water, and food as the main three elements. I have here the kilograms, the mass per person per day. It's average values. We all don't breathe the same. Some water requirements might also differ if you are in a very desert, hot and dry area and doing a lot of sport. So those are only average values, but they serve us as a reference to get an idea how much consumables we would need as humans in order to survive. And at the same time that we are taking these three things, we are producing waste in form of carbon dioxide that goes through the atmosphere, and then also liquid and solid waste. Liquid comes out of our skin to our atmosphere or in form of sweat, urine and feces, and at the same time we'll also be producing heat. Beyond that, for surviving, we could argue that we also need hygiene water to clean our body. It's not the same urgency as our drinking water, but of course, if we don't have any kind of hygiene within our body, we may also end up having healthy issues. So we take these numbers, we know what we need, and we calculate. We want to go from here to Mars and back, one family, group of 4, take your best 3 friends. During that time, going and coming back safely, we would require 3 tons of oxygen. We would consume 3 tons of oxygen, about 2.5 tons of food. And the biggest problem is the big amount of water that we will require for that. So it depends how much water you take for hygiene purposes, but we're talking between 13 and 37 tons of water that would be consumed. And if you put all of this together, it's a big mass. We would need a lot of rockets to bring this up. And we're only talking about the oxygen, the food, and the water. We still don't have our habitat, we don't have our experiments, we don't have our astronauts, we don't have our radiation protection. It's only the consumables that we need to survive. That is not feasible. I always put a number in euros, but I don't trust that anymore, so I'll just keep the number. When we plan a trip to Mars, we'll plan it in a similar way as you probably plan your holidays. If you're going hiking to the mountains for a weekend, you take literally your house with you. You take your tent in your back, you have your clothes, you have your food. Depending where you are, you might be also taking your drinking water for the 2 days you're out. So it's pretty much everything on your back, on your shoulders. But if you're going for a semester out, I'm sure many of you have or are planning during your PhD, spend some months out, You pack differently. You pack some clothes and you assume wherever I'm going, there's going to be a washing machine, there's going to be water, so I'll be able to wash my clothes. I don't have to take food for 6 months. I'll take money with me and buy food there. So we're talking about recycling and the use of resources in situ. And in the same way, we would plan our human spaceflight mission. If it's short, it's okay. We can deal with that and take things with us. If it's long, we'll need to learn how to recycle or use the things we will find wherever we're going. And that's shown here in the next slide. So when the mission is short, and the weekend example would be in this case the lunar missions, they lasted about 2 weeks. And that's still short enough that we just take all the fresh inputs we need and all the outputs. Of course, we just don't throw them away on the lunar surface in any way, but we pack them and they are disposed. And that would be what we call, in terms of human spaceflight, an open life support system. The other extreme would be the city on Mars. If we want to have, or we plan to have at some point, 1 million people living on Mars, they will not continuously get resupplies from Earth. That's not sustainable in any way. So somehow they will have to either depend on the resources on Mars or have a fully closed system where they are able to recycle from all the human waste that we produce, all the fresh new resources, oxygen, food, and water. Today, we are somewhere in the middle. In the International Space Station, we have a system that you can call it partially closed, partially open, where some of the inputs are regenerated and some of the inputs are fresh, brought from Earth. And how is that done, this regeneration of what humans need? Well, clear example here on Earth. You all recall, surely, your biology lectures at school. We have plants, they do photosynthesis, they take the carbon dioxide, they produce oxygen. They also participate in the water recycling on Earth, and on Earth you have these water cycles. We have clouds, it rains, goes to the rivers, and so on. So we have natural cycles on Earth that close the oxygen cycle, the water cycle, and what we can relate to the food, the carbon cycle, among many other material cycles being closed here on Earth. But biology does already do that. Beyond that, we can use what we call physicochemical technology, so based on physical or chemical principles. And with that, we're able to recycle already oxygen and water. We do that here on Earth already. Water recycling systems exist everywhere around us. Quite many houses have reverse osmosis to purify water for drinking purposes. Oxygen production, as you surely also know, can be produced by splitting water molecules, so we can obtain oxygen and hydrogen on the process. So these processes are also already known. Now, for the first missions, all those things were not too important. The first missions were very short, and as we have seen, also the lunar missions were considered short. So when they were going up at the beginning for a day or two, I was okay. They were just taking everything with them. But we've gone a long way since then, and we're nowadays here. I always only showed the picture of the ISS. We also have the Chinese station up there at the moment. So we have those two stations up there. And on the ISS specifically, it's possible on average to recover 42% of the oxygen from the carbon dioxide that the astronauts exhale. We're able to recycle, I think it's even a little bit more now, over 90% of the water that is used on the station. So only a very small percentage has to still be refurbished from Earth. And the biggest problem right now at the station is there's 0% food production. There have been some experiments, and I'll show a couple of pictures later on. There's been a salad already on the station, but not as main part of the life support system. Those were small experiments taking place on the station. And I'll have a short video to talk about the recycling on the station.
NASA urine recycler, test 1, sample 1. It's the first water we've got out of the machine. This will allow astronauts to recycle their urine and sweat into beautiful, clean, clear drinking water. First sample, transparency, subpar. Oh, that's just piss. That's just straight up piss. Smell, urine-y, and damn it! Oh, that's piss. I don't know why I have to drink it. It's piss. It's yellow. That's piss all the way down to the bottom. Results have not been promising. Edison, you know, it took him like thousands of tries to find the right filament for a light bulb. And maybe we're being idiots, or maybe we're... that's piss! Let's just see who drinks it. I'm not gonna drink it. I can sit here all fucking day. You drink it! Do we even have a machine? Are we just pissing into this bottle? That would go great with a cheeseburger if you like drinking piss with your cheeseburger. Hey, that's not bad.
Like a glass of water that a friend pissed a little bit into.
We made water! Somebody give me a glass of piss. I want to celebrate.
So that's quite an old video and obviously a joke. But yes, the process, not like that, but that happened. There's a machine on the ISS that's able to recycle the urine from the astronauts, but it's not the only one we have on the station. But we have, I'll explain the graphic in a second. The technologies that we have on the station are physicochemical technologies, so the ones based on physical and chemical, and not the biology as we have seen the differentiation before. So on the station, we have our astronauts. It's an old picture. That time there were 6 usually on the ISS. Currently it's usually 7. There are also a couple of mice there that have, let's say, similar needs to the humans. And we have all the systems and cycles that we need around them, starting with the air. We have the air is first being adapted or changed for temperature and humidity, and adapted if necessary, regulated if necessary, temperature and humidity-wise. This air is then brought to a system that removes the carbon dioxide. We don't need to do that actively here on rooms. We probably have some kind of air circulation system, but we can also just open the windows and get fresh air from the outside. On a space station in a closed volume, in the middle of nowhere, or in the middle of vacuum, our atmosphere will continually accumulate the CO2 that the astronauts exhale. And if we don't do anything, that would reach a point where it would be toxic for the human. So it's very important that we have some kind of filtering system that actively removes this CO2. The air with less CO2 or with almost no CO2 then returns, and it's the air that comes back to the cabin. The CO2 can then be further processed. As we have seen before, part of the oxygen comes from the CO2. So there's a system that processes the carbon dioxide. And in this case, the system on the ISS produces water. And then this water is electrolyzed, so split it into hydrogen and oxygen to produce oxygen to refurbish the atmosphere. Same as with the carbon dioxide here, we don't need to push extra oxygen in our rooms. We have enough outside, we open the windows and we get fresh oxygen. But on the station, they would keep consuming the oxygen, and at some point it would reach a level that it's not breathable for the humans anymore. We might need to add a little bit of nitrogen in our atmosphere, and that's not because we humans consume nitrogen in any way, we don't. But we are in a closed volume in a space station with vacuum outside, and there's definitely going to be leakage from our atmosphere. We'll be losing a very tiny, small of our atmosphere, but it's, I would say, impossible to have it completely 0% leakage, so a little bit of nitrogen might need to be added to keep the pressure inside the station. From the water side, as we have seen, urine recycling is a thing, was long tested on Earth by NASA and on the station working. So the urine is collected and water is produced or recycled from the urine. And with that, the astronauts have enough water for drinking. I don't know why NASA put shower here. There's no showers on the ISS. We don't have gravity on the ISS, so they cannot just go under a shower and clean like that. They actually use water with towels to clean their body surfaces. And at the same time, we also get the water, of course, from the condensate on the atmosphere. As I've said before, we also keep bringing water to our atmosphere, and that's also collected and recycled to potable water levels. The waste that we humans produce, the solid waste, is just disposed. I'm not sure if some of you have seen The Martian, the movie The Martian. If not, highly recommended. There's potential use for the human waste in the future to cultivate potatoes. We'll see a little bit of that later on. And it's not showing this graphic, but the food is all brought from Earth, so there could be here an extra box with food going into the astronauts. And that's the main structure of the life support system on the ISS. I brought a couple of pictures and names of the technologies that are actually currently being used. For the water systems, there are 2 technologies. One is the multifiltration that takes care of the wastewater. It's a system of different types of filters, from particle filters to activate charcoal and ion exchange filters, so different types of filters that make sure that the water gets clean of any contaminants. And particularly for the urine, it's a different process. For urine, urine is much more complex than any other wastewater that is produced for hygiene purposes or for condensate. And it has several elements that we cannot just easily treat. And for that, we need a distillation process. So we kind of easily set how to heat it up, the urine, and let the water condensate at the surfaces and then collect this pure water for us to separate the water from the rest of the elements that are present in the urine. And that is a little complicated in space because distillation comes with the principle that hot vapor will raise up and will then collect this vapor in certain surfaces at the right temperatures. But in microgravity, vapor doesn't go up. So they had to design a quite complex system that rotates to create, let's say, artificial gravity for the system, so the distillation process could take place. So it's a system that rotates, which creates a lot of problems on itself, mechanical parts that can break more easily due to the rotation, or also perturbances on microgravity, which having humans, it's anyway not a big deal because you also have the humans moving inside the station. But yeah, those are the technologies used. And what you see on the right are two racks. One rack is, I'm not sure about the right size, but a little higher than a person. So we have two big wardrobes to recycle this 90% of the water for, let's say, 7 astronauts. For the air, we have the oxygen production part and the carbon dioxide removal and reduction. So what we have seen here on these parts. So for the oxygen production, there's the technology used by the Russian side and the American side. They use the same principle. It's all based on water electrolysis. Some of you might be working not for human spaceflight, but on water electrolysis for other purposes, for space propulsion or so on. And those are the two technologies used. For the carbon dioxide removal, there's here Bostuk and Sidra, the carbon dioxide removal assembly. Those are, again, the Russian and the American technologies. And they are basically some kind of material that at the right temperature and the right conditions of humidity, CO2 particles can get trapped in. And when we change this temperature, humidity conditions, and maybe apply some under pressure, the CO2 particles can be extracted. So it's a filter that we can fill in with CO2, and when we want, release the CO2 and store it and further process it. And this further processing is important because we want to regain the oxygen, which is done by a CO2 reduction with a Sabatier reactor, which is the picture you have on the bottom. Sabatier reactor combines, or we react carbon dioxide with hydrogen, and with that we produce water that then we can get the oxygen out of by water electrolysis, and it also produces methane. This methane at the moment is not used. It's been extracted from the ISS, it's been vented, but this potentially could be used also for propulsion purposes, or we could reuse it further to regain the hydrogen, since for the Sabatier reactor we need hydrogen constantly, so we would somehow close the hydrogen cycle in that sense. Now, those are all American and Russian technologies on the station. Have you ever heard about European life support systems technologies on the station? Do you think there has been one? Are we Europeans capable of that? So the answer is yes and no. There's the German expression Jein, which is yes and no at the same time. There has been a technology up on the station. I think it was brought in 2018, and it's been decommissioned now, which was a technology demonstrator. So it was not really part of the functional life support system of the ISS, but a technology demonstrator. So it was an ESA-funded project. The prime building the hardware was Airbus in Germany, in Friedrichshafen. And it was capable, or it was meant to be capable, of providing enough for 3 astronauts. So I have the 3 astronauts here. It had a system also to remove carbon dioxide. Since we produce 1 kilogram per person per day, it was able to remove 3 kilograms per day. Then again, we have the same technologies we have seen. So there was a Sabatier reactor and water electrolysis in order to close the cycle from the carbon dioxide, get enough oxygen for 3 astronauts. There is a little bit of water coming extra from the outside and the methane that is vented, because we need to balance, so it's not fully closed. But that was up on the station. They made they managed to make the CO2 removal system work, the electrolyzer as well. They had several problems with the Sabatier, and I don't think it ever worked as a full system, but there's like in many space missions, a lot of lessons learned on how to do things differently in the future or improve systems in the future. But that's the major project we had from European side on life support system on the ISS. So with problems, but still a lot of lessons learned on that. And it's being decommissioned now. They stopped the operation officially, I think, 4 or 5 months ago. Now, on the station, they get the food from Earth, and I usually show a couple of pictures. That's, I think, food from, I'm not sure if it's from the ISS or still from the space shuttle. But I always like to explain, I had the opportunity to meet the company that was producing the food from one of the German astronauts. That was the company Lufthansa Service. And they basically had food in a can. I'm not sure how good you can recognize the picture here. There's a can floating. And I also found it interesting that there's a different concept on how to produce food for astronauts. From the American side, because they have a lot of astronauts, they have a catalog with different foods in cans, in bags, and the astronauts can pick which things they like, which they don't, and then someone sits and makes sure that they have a balanced diet, so they provide them with their diet for the, 100 days that they will be on the station. For the Europeans, because we have only a few, they have, at least these last years, every time, selected a chef from the country and have made a big event out of it, producing the food for that specific astronaut. So there has been a lot of very regional specific food in space. I lived for many years in Stuttgart, and one of the German astronauts was from the region. So many of the food you get in the Stuttgart region, if you've ever been there around, there's something called Kässpätzle, and he had this Kässpätzle up on the station. So it was very tailored for each astronaut. I guess with time, if we keep getting more and more astronauts, that's not something that's going to continue. But for many years, that has been the case for the European astronauts. I've not mentioned that the food is slightly different than what we eat here on Earth, because on space our body changes, and one of the things that happen is that because there's no gravity, the fluids of our body tend to go up. If you look pictures from astronauts compared to Earth, they tend to have a more puffy face and the legs more tiny. We call them chicken legs and puffy face effect. And one of the effects that has is also that we don't smell that good anymore. Our sense of smell is reduced. And part of the taste when you eat actually comes from the smell. So what they had to do for the food of the astronauts to make it more tasty is to put more salt, more pepper, more things on it, compared to what we would usually eat here on Earth. And that has nothing to do with the taste itself, but with the smell. So yeah, humans in space, it's a complicated topic. There are many aspects to consider. From the waste, it's at the moment just all being put together, not only the human waste, but from experiments and spare parts when we repair things and things like that. And it's being burned during re-entry in the atmosphere. Now, that's the current status. That's where we are with the ISS nowadays. And the goal of our research is to make it possible to one day have that big city on Mars for a lot of people. And as we have already seen, that implies recycling a lot. And we have seen that nowadays we manage water and oxygen, especially water quite well, but oxygen as well, with these physicochemical technologies. But we cannot manage that city on Mars if we don't go into biology. For long duration future space missions, not today for the ISS, but looking farther away from Earth, we will need this biology. And that's not a new topic of research. That has been already for many years since we started going to space, actually. And I have a couple of examples of experiments that are happening or have happened some years ago here on Earth. Has anyone heard about the biosphere too? Couple, okay. So that's a huge building. I don't know if there's a good reference here. I think there's a small golf car on the side, and you see the trees. So it's huge. It's about 16,000 square meter surface. It's a sealed environment. It has its own atmosphere, completely separated from the outside. And it has different regions, one with the jungle, another with a more desert area. So Earth is represented somehow by different rooms, different compartments there. That was built during the 80s, late 80s. And there were 2 experiments in there. One lasting for 2 years with 8 people inside. It's quite big. It has nothing to do with the size of the ISS. They really had place to run and a lot of work to do with the vegetation, with this huge area of vegetation. But the idea is to study a closed environment. So we have Earth put in a box and see what happens with humans on it as well. It's called Biosphere 2. There was not a building called Biosphere 1. Biosphere 1 is our Earth, and they built a second Earth on Earth, and that's why it was called Biosphere 2. The second experiment was 6 months, and it ended because of, I could say, political reasons. The management people disagreed, and one of them just went and opened the door. And if you're doing an experiment with a closed system and someone opens the door, it's not closed anymore, so experiment officially ended. During the first one that lasted 2 years, we learned a lot of things. For me, the most unexpected one, because as an engineer, would never thought about it. In such a building, you don't have wind. And trees apparently need wind. If there's no wind, their structures don't grow as strong. They grow strong because they have to fight the wind. That's something that, as an aerospace engineer, is the last thing I would have thought. One of the biggest problems they had was that the oxygen in the atmosphere decreased. And I always ask these questions to my students. Why do you think the oxygen decreased in a building full of plants? I know you're probably all engineers, so not biologists, plant, but it. So the problem is the plants. My engineers always blame the plants. The plants were not producing enough oxygen. It's like, yes, that's true, but it was not their fault. The problem was that the material with which the building, the concrete with which the building was built, was capturing CO2. So the CO2 that the humans were producing, or the plants during night were also producing, was being captured by the building itself. So it was more a material selection or material processing that made it then not possible for the system to have enough CO2 to produce enough oxygen. They solved the problem by injecting a little bit of oxygen, so they were cheating. It was not a fully closed system for the 2 years. But of course, if you document that, it's still okay to continue with the experiment. But they have many more other problems. They had insects and small animals in there, and some insects overpopulated, other completely disappeared. So for me, the main lesson learned from such an experiment is our Earth is very complex. We have big buffers. We have a huge atmosphere. I know how many tons and tons of air we have around us compared to what they had in there. It's a very small percentage. We have big oceans that are also used as buffers in our case. They had a small, I call it swimming pool, they call it ocean. It was a big amount of water, but of course, nothing to do with the size of our oceans. So reproducing Earth in a closed environment, it's very tricky because it's very easy that it goes out of balance. We know that here on Earth also our Earth conditions are changing and the CO2 in the atmosphere is growing up, but the same process in here happens within hours probably compared to what we are observing over years and years here on Earth. So it's very unlikely that we just go to Mars, make a big greenhouse and put a bunch of things from Earth and we just hope we can live there. But that does not mean that we cannot use biology for our purposes. That's why I like to talk about another experiment that has already been ongoing for, I think, 30 years now. It's organized by ESA, but there's many partners from Europe-wide, and there's one of the big partners is in Barcelona surrounding. So if you ever get the chance to go to the Universidad Autónoma, there's a pilot plan of the project Melissa. And the idea here, I'll just go down. The idea here is to use biology, but in a more controlled way. And for that, we have compartments. There's compartment 1, 2, 3, and 4. And I'll not go into the details because that's biology and it's also not my topic. But we have one type of bacteria in compartment 1, a different type of bacteria in compartment 2, and another one in 3. And only one type in each compartment. What we do here is select them wisely. So we make sure that the first compartment, the fixed bacteria, the thermophilic anaerobic bacteria, are able to take the solid waste from the humans and produce certain products. That these products are what my next bacteria need to produce the next outcome. And I do that in a chain up to a point where my last bacteria produces the nutrients that my plants need to grow. Since I only have one type of living organism in these compartments, I can study very well how these bacteria work, what happens if I put more light, if I reduce the temperature, and I can control them very accurately. So it's not more like, let's put nature together and let's see how they interact, but rather making sure that we control things in a very controlled, exact, precise way. That might still come with problems. It might still be that because we're simplifying things, nature is very complex here on Earth, and we humans are also very complex, and we need a lot of things for our food and a lot of trace elements and things like that. So I don't know if long-term that could at some point be an issue, but it's all about then controlling which elements I'm taking in my last compartment where I have the plants, or I'll talk also about microalgae, which at the end are the ones that are eaten by the crew. So we can still play with that. But the idea is, keep them separate so we can learn them properly, study them properly, and control them. Beyond those projects, there are other things going on, and because I'm living in Germany, I tend to explain the German things. Because I collaborate with them a lot. So one of the projects that I also found very interesting was Eden ISS. It was an European-funded project guided by or led by the German Space Agency, but with different partners worldwide. And they had 2 big containers, which they sealed as good as they could. And they did not bring them to space, but to the Antarctica, to an isolated environment. And there they tried to study how to cultivate plants in an efficient way in an isolated environment. And the guy that was taking care of that for a year is a colleague of mine. He was not living here. He was living in a station a couple of kilometers away. And to go there during the winter in the Antarctica, he had to suit up, not like an astronaut, but close, walk all the way to this greenhouse. And he had not a nail lock like in a station, but also a similar thing where he had to get rid of all the equipment. So I think for him was a little bit of an astronaut feeling, at least, being there. And they collected a lot of knowledge already on how to grow the plants more efficiently. And I think the people living on the Antarctica, beyond the guy that was taking care of that, it represented a high psychological value for them to, you're close for several months on a station with probably all the food also in cans or packed. And every now and then you get some fresh food that is being produced in situ. And that, of course, after weeks and weeks of isolation has a very positive psychological aspect. These containers have been back to Germany now for a while, and they're going to be brought to another facility, which is situated in Cologne, where the astronaut center is. They built there a hall that represents or mimics the lunar surface, so they can train their EVA activities. They have an offloading system to simulate the gravity of the moon. And next to that, there's a habitat. And next to the habitat, they will put this greenhouse. So it will also be possible to simulate a lunar base and the cultivation of plants in a lunar base, of course, under Earth gravity. So there are things we cannot replicate, but at least part of the experience can be replicated with those experiments. Now, those were all experiments on Earth. And I brought also a couple of examples of experiments in space, because biology has also been experimented already in space. Those are already all pictures from systems on the station, which allow to cultivate plants in small sizes. So I would say the experiments taking place so far have been of the size of a microwave. There has been salad, which has then been eaten by the astronauts and other type of plants, to see how we can, how plants can grow in microgravity. Plants on Earth feel gravity and they grow accordingly to gravity. So not having this gravity affects the way plants grow, but we also want to see how we can have that in a controlled environment. We have the atmosphere of our astronauts and we need our small piece of equipment. It's not like here on Earth that plants grow on the open in our Earth atmosphere, but we can also control a lot of the parameters on which these plants grow. Now, those are what we would call higher plants. And my field of research goes into a slightly different topic with its microalgae. Microalgae are algae that are very tiny, that you have to look to them through a microscope. So those cells here are micrometers, 4 to 10 micrometers diameter. And they are, so when I start talking about plants, I manage now at my house, but I used to tell my students that I always kill plants at my house. I don't know how if you guys manage to keep them alive in your house, but I always worried they don't have enough water and put more water, and they end up probably suffocated because they cannot breathe anymore because of too much water. The nice thing with algae is that they are much more simpler than that. And because they are simpler, it's easier for them, or they are more robust against people like me that doesn't know how much water they might need or not. And that's why it's one potential candidate for producing oxygen. They also do photosynthesis and food on a space station or on a lunar base. The question might be how good they taste or not. I unfortunately forgot to take mine with me when I left yesterday Munich. They are very salty. They smell like the feed that you give to the fish. So yeah, they might not be the most tasty food that we can produce in space, but it's very efficient in terms if it requires less resources than plants, less space, less water, less light, and so on. And being simpler, it's also easier for us to understand how it works and not to kill it on the process of cultivating. Now, I'm an engineer. I'm very much into the topic microalgae for space applications because as an engineer, we also have a lot of work to do on that. We need to understand the biology and what the algae needs and what happens when we change temperatures and things like that. But we also need to be able to design the equipment that for a lunar base is able to cultivate these algae for long periods of time at the right temperature, providing the right light. So there's also a lot of engineering that has to happen around that. And a good example of that is an experiment we brought to the ISS in 2019, which was a photobioreactor where we cultivated our algae. So that's a chamber where the algae were flowing. That's open after we finished the experiment. And we had our algae sample taken from our equipment after a couple of weeks. So that picture you see on the right are microalgae, Chlorella vulgaris, cultivated on the station for 2 weeks. There are several challenges that you might also face if you're working with microgravity purposes in the space area. One of the big problems we have is algae live in a liquid form and they produce gas, or we want them to produce oxygen in a gas form. So liquid and gas separation in microgravity is one of the big challenges that we face for that, but in many other fields of space research is also the case. Now, I did that when I was working in Stuttgart. I recently moved to Munich and I'm trying to continue this research. We got a huge building. I promise I'll show you a picture of the lab. We have a huge facility that was built to cultivate algae for not space applications, for biofuel production together with Airbus. And now we are using it and we're planning that maybe in one of there's 3 big areas, the 3 big doors, that we can simulate a lunar base photobioreactor. So really building the system at the size we would need for a lunar base, which is probably going to be a couple of thousands of liters, and actually test the operations of a system as they would be in a lunar base. So I'll probably have a couple of my students not living there as in a lunar base, but at least daily taking care of the system as it would be on a base. And yeah, we are still far away from anything that might happen one day here on Mars, as I've mentioned at the beginning. But again, we are doing the first steps onto that direction, and that's why we're focusing our research on all these areas. I'm going to finish my talk with a short message I feel always I need to do when I talk to the general public. Not so sure this is the case here. But before the question comes, and you might have been confronted with that question working on the space sector, it's like, but why do we do that? Why do we spend so much money researching on space? Don't we have enough problems here on Earth? And I like to explain that what we're doing precisely, this life support system research, for one day being able to live on Mars is something that it's helping us already today here on Earth. If we want to imagine how we would live in such a harsh environment and develop the right technologies for that, those technologies can also be used here on Earth. I've been talking today about CO2 and processing CO2, problem we have here on Earth. We look a lot on recycling water and have a meaningful use of our water. It's also something we might need in some areas here on Earth. And we're looking at systems to produce food in an efficient way. I mentioned that algae are particularly efficient. but also other means of cultivating plants, things that we're also by now doing here on Earth with aquaponics, hydroponics, and so on. All those things are also applicable here on Earth. So I always end my talk like this, so I avoid having the first question on that direction. But having said that, I really hope that one day we'll see one city like that, or maybe your children will see a city like that in the future. Yeah, I'm really happy that I had the opportunity the last years, first from Stuttgart and now from this professorship in Munich, the opportunity to work in this area. I guess you can tell that I'm really fascinated about my work and what I do. I'm very happy to have any discussions with you guys or try to answer any questions if you have. I'm here all day or for the next hours at least. So thank you all for having joined today. I think that probably none of you are really working on this area from what I've been told that is happening here at the university. But if you're interested, you have my contact data now or anytime. Just, yeah, let me know. Thank you.
All right. Thank you, Gisela, for your talk. Time for questions. We have time for a couple of questions. Who wants to start? Andres?
Because of your expertise, I mean, I'm very loud.
Yes, but just for the...
Okay. Because of your expertise and your focus, you have looked at the biological part, the consumption of oxygen, the production of solid material, feces, and things like this. But I was curious when you saw the big equipment that they use to recycle the urine, and the numbers are outstanding, 90% recovery of water. But what is the energy consumption of that thing? Does the ISS have sufficient solar panels? I mean, it has to be because it's doing it, but are they taking batteries or some way of energy? Will it be applicable on Earth to recycle our waste to produce water since we have a problem?
Energy, it's a very good point. On the ISS, they have enough energy to recycle urine. If they would have enough energy to have enough artificial light for plants if required, that would be the next step, but probably not. But on the ISS, there's an average power consumption of 110 kilowatts provided by solar panels, and there are batteries for the night part. And only a tiny part of that is the life support system. There's also a lot of experiments that use a lot of power. So it is not yet an issue, but if we start talking about this using plants, we would probably need over 100 square meters per person of cultivation area for a human. And if we are underground on Mars to be protected from radiation and we want to use artificial light, that is a lot of power. Also for our microalgae, we're using a lot of power. And that with Earth, it's a very good point, because for me, it's exactly the contrary. Here on Earth, we have a lot of surface where we can capture sunlight. And the example of the algae is a very good example. We make it very compact, and it requires a lot of power. But we are willing to use this power or pay for this power because we need the oxygen. I cannot survive with the oxygen. I have to put power. If it's not to produce oxygen with the algae, I'll need to split water via electrolysis, so the power has to be there. We were asked for a project if we could use our knowledge for Earth applications, and we came to the conclusion that makes no sense. Here on Earth, you can be bigger systems of algae that are spread more in the surface, like in a swimming pool, and they all get sunlight. We don't need to use all these LED panels and be as efficient as we are. So for me, the thought was the other way around. Like here on Earth, we have enough sun. We can get enough power with the right surface because we have also a lot of surface compared to what we have on a station.
Follow.
I can see that from the algae, but I was thinking more on in Spain we have a drought in the last few years, it rained a lot and it's better. But we didn't even implement desalination plants because of the power required to do the desalination. So I was thinking here with the urine the same. If it's really time consuming, we won't be able to recycle our things or apply many of these technologies.
I don't think that's less power than what would be the other options that have been considered here on Earth. We might come to a point we will have to require this energy. Same as with the oxygen, it might be that here in Spain at some point we have to say, okay, we need the water, we need somehow to use this, to get this energy for that. I don't think that would be less energy consuming, no.
Mario? Well,
Thank you, Gisela. So very, very interesting topic and have a lot of curiosities about it. What do you think is the most challenging part of the, what's it called, a bioreactor for the algae? I guess when you have the sunlight, you probably need to control the temperatures or heat rejection if it's being generated heat, managing the salinity. I don't know, what is the element from an engineering standpoint that is the most challenging one to design?
So there are two things. One is, if we talk about microgravity, the separation of gas and liquid is the nightmare that we have to face. And again, that's not a problem only for photobioreactors, but in many fields that microgravity becomes a nightmare. But we are also focusing on lunar surface and Martian surface where we don't have that problem anymore. And we have less gravity, but still gravity. So liquid gas separation stops being a problem. And then we focus on other things. And for me, the big challenge that we need to solve, I don't think technically there's anything we cannot achieve. But we are working now with me and all other groups around the globe with small lab photobioreactors. Processed by lab personnel that knows what they are doing very well and they have the right environment there. If we want to have something like this, the systems will not be 1 liter photobioreactors anymore, but we'll have something huge. And then all the problems magnify. We might have contamination problems. How are we going to deal with the fact that there will be contamination in my system at some point? If I'm running it for 10 years instead of in a base and not for 1 month in a lab environment, I'll have to deal with that. I might need to have my system modular so I can deal with that. So designing the system and the operational part of it will also not bring thousands of liters of algae cultivation to the moon. We'll probably bring small bag of algae and a lot of water and grow up the algae there. Those things, we have not really considered them yet. So it's not a big technical challenge, but it's something we need to investigate. We'll find a way to do it, but it has not been done yet.
How robust is the system in the sense that this population of algae, maybe you tweak a setting and suddenly the population grows and explodes and then everything dies, or it dies off? And how robust is?
Depends on the algae. There are thousands of types of algae, and there's algae that are very picky and others are less. The one we have selected for our experiments, after I explain how bad I am with plants, is one of the robust ones, which is also what you want in such a base. Yeah. We selected an algae that can live with wide range of temperatures, of CO2, of pH values, that it's actually, robust in that sense. Biologists try to usually like to work more with other types of algae and have their specific parameters. As an engineers, we try to tell the biologists, no, give me the algae that it's okay, because I'll have a thermal control system, but it will be not precise to the 0.1 Celsius. I might need some margin. So we look for the, we try to find each other, biology and technology, in a middle point somewhere.
Okay, thank you.
I think, unless I don't see any other question, I think we're going to close the session because it's already 2 p.m. Okay, so thanks very much,
Thank
Gisela.
you. Thank you all for coming.
Thanks. Okay, so thank you.